In this video, we're going to discuss the different types of plasticity responses that can occur in response to damage to the nervous system. As we've discussed previously, brain plasticity is this amazing ability of the brain to change. We can see it in learning, we see it in response to addiction, and what we're talking about here is in response to damage to the nervous system. Now, in a previous video, we talked about different types of brain damage that could lead to a plasticity response. But keep in mind, this plasticity response happens in both the central and the peripheral nervous system. And again, following brain damage or any damage to the nervous system, the surviving areas, they want to either increase or reorganize their activity. They want to replace any lost function. They want that nervous system to continue to operate efficiently and effectively. So we're going to talk about two different types of mechanisms that aid in this recovery of function after damage to the nervous system. First, we'll discuss the regrowth of axons, and then we'll discuss axon sprouting. And keep in mind the type of response that occurs depends on the part of the nervous system that's damaged, how severe that damage is, and whether or not the cell body of that neuron survives. So the first type of response we're going to talk about is when the cell body survives. So there's some sort of damage to the nervous system, and that cell body survives. So this is going to most commonly happen in injuries to either your spinal cord or out in the periphery, in your peripheral nervous system, right? Like say something crushes your arm or a car rolls over your foot. So this type of plasticity that we're going to talk about is the regrowth of axons. Now again, this type of plasticity is most commonly going to occur when the cell body survives. However, it's going to depend on the severity of that damage and the location of that damage. The idea is that damaged axons do grow back under certain circumstances. So in the peripheral nervous system, a crushed nerve might be able to follow its myelin sheath back to the target. The myelin acts as a guide, and that's what you can see here. And remember, in the peripheral nervous system, the cells that make up that myelin sheath that we see here are Schwann cells. What we've talked about previously were oligodendrocytes. So don't let that confuse you. So this is your cell body, this is your axon, and it's surrounded by that myelin sheath. Here's that site of injury, and that part of the axon is going to degenerate. But you can see there's still left here some of that myelin. And so now that the proximal end of the injured nerve fiber, that axon, can follow that myelin sheath back to its original target. And here that target's the skeletal muscle fiber. It could be something else in other parts of the nervous system, perhaps. And so now it's grown, it's reestablished that connection, and function has been reestablished. So again, this is in the peripheral nervous system. Remember, outside the brain and the spinal cord, all those nerves going out to your muscles, all those nerves bringing sensory information back into the CNS. And so again, if there was some sort of crush where the axon wasn't severed, it was only damage, you could have this regrowth following that myelin sheath. Now, a severed or a cut axon may not grow back. So let's say there was a deep cut to your arm or a deep cut to your leg, and some of those nerves were damaged. The same idea could happen. That axon can regrow, but if it doesn't have that myelin sheath to follow it back to the correct location, it may not grow back to the right location. And then functional recovery could be a little bit iffy. Now, in the central nervous system, so in our brain and our spinal cord, especially the mammalian central nervous system, because we're mammals, right? So that's what we're going to talk about. We don't see the regrowth of axons. We don't see axons regenerating. And this diagram here shows the main differences between the central nervous system and the peripheral nervous system. So here we saw what we saw in the previous slide where you have your injury. Macrophages are going to come and they're going to gobble up that dying debris, but then that nerve can follow its axon back to the original location. In the central nervous system, we have that injury, but there's no macrophages to clear up that debris. We don't have macrophages in the central nervous system. We have microglia, and they operate a little bit differently. But we also have astrocytes, and so what scientists have found is that these astrocytes come to the location of that damage, and they act as scar tissue. They gobble up some of that that debris, but they also release some chemicals that inhibit growth. So in the central nervous system, the question is, why don't axons regenerate? Why don't they regrow? And that's kind of the million-dollar question that science wants to answer, because if we can figure out, and we know why. So we know that nothing clears debris. We don't have those macrophages. We know that the astrocytes act as a scar tissue. They create what's called a glial scar. And that these glial cells will also release some chemicals that inhibit growth. Some of them are named something like, as creative as no-go was one of the chemicals that's released. So a lot of work by scientists is aimed at trying to figure out how can we alter these things in the central nervous system? What can we do to clear this debris? What can we do to break up that scar tissue? I mean, some scientists suggest that scar tissue is actually good as well. And so again, this is just showing you that no debris is cleared. We have that glial scar and some of these chemicals, these inhibitory molecules that those glia release. So when the cell body doesn't survive, what we see is axon sprouting. And this is sprouting of surviving neurons' axons. So we also can call those collateral sprouts. So the idea is that new axonal branches are formed by non-damaged axons. And remember, a cell only has one axon. So those non-damaged axons have to come from different neurons. So that's what we see here. So you have neuron A connecting to this other neuron B connecting to this other neuron, and it's been severed. This axon's been severed for some reason, somehow, or damaged. So that connection deteriorates. As that deteriorates, this neuron, cell body of neuron A, is going to die. It's going to degenerate. It's not going to be in there more because it's, again, you can't have dendrites in a cell body hanging out with nothing else there. So that's going to go away. But we want to have recovery of function. The brain really wants that plasticity response to occur. So what happens is neuron B, the surviving neuron, its axons sprout. So you can see this axon sprouting here, the axon terminals sprouting, all to fill in this neuron here. So in the beginning, A was connected to A, B was connected to B. Now B is connected to the receiving neuron A and B. And here's another diagram demonstrating this axonal sprouting or collateral sprouting. So here you have axon 1 connecting to dendrites of one neuron. So now we have just one receiving neuron, and axon 2. Something happens. Axon 2, the neuron dies, the axon is injured, it degenerates, and so now this cell, this neuron's gone. Well, we want to recover function, and so axon 1, these axon terminals are going to sprout. And again, that's called collateral sprouting. It goes in and it innervates where the other axon was previously. So now we have the same number of connections innervating this receiving neuron. Now, as I said previously, this plasticity response decreases with age. And that was one of the things I was studying during my PhD program. We wanted to know why this decreased with age, if there was a way to maintain plasticity over time as the brain ages. And we had some promising animal models that we worked with. But basically, it all boils down to diet and exercise. And so that's what I'll leave you with. Eat a diet in rich leafy greens and colorful vegetables and fruits because they have all sorts of antioxidants that are good for the brain, and get regular exercise.